Chronic myeloid leukemia. Cells communicate using chemical signals. These signals are proteins made by the cell that are released into extracellular space where they move to neighboring cells. Signaling molecules are called ligands and are only accepted into another cell if the cell possesses a receptor. When the ligand meets the receptor, a change is triggered inside the cell. Local signaling includes paracrine and synaptic signaling. Paracrine signaling is when cells communicate through chemical signals. These are done with neighboring cells. Synaptic signaling is when a nerve cell releases neurotransmitters molecules into a synapse, stimulating the target cell. Long-distance signaling is done through hormonal signaling. This is when certain endocrine cells secrete hormones into the bloodstream and then are received by any cell in the body. A signal transduction pathway is a set of chemical reactions in a cell that occurs when a molecule attaches to a receptor on the cell membrane. The pathway is the series of biochemical reactions inside the cell that reaches the specific molecule. After reaching its target molecule, the cell generates a response. A ligand is a signaling molecule that binds to the receptor of its target cell. The message carried by a ligand is relayed through a chain of chemical messengers inside the cell to influence the activities inside. Calcium ions act as a second messenger in the process of signal transduction. Calcium ions regulate cytosolic levels in the cell during the process, as well as balancing the cytoplasmic concentration of Ca2 ions. Cyclic AMP is another kind of second messenger used in the process of signal transduction. Adenylate cyclase converts ATP into CAMP. When a cell receives a signal, its processes depend on the type of message received. Certain signals are responsible for opening or closing a channel, amplifying a signal, relaying the signal to other portions of the cell with signal transduction, or activating or inactivating a protein enzyme. Protein kinase is a type of enzyme that transfers the ATP phosphate groups to proteins. It occurs in a signal transduction pathway and results in a shape change that initiates each enzyme. Once epinephrine is released into the bloodstream during a flight or fight response, ATP breaks down epinephrine into blood glucose. A protein-stimulating adenylate cyclase is released as well, allowing for the quick release of glucose needed into the blood. Tyrosine kinase receptors regulate normal cell processes and play a critical role in the development and progression of numerous cancer types. Mutations in receptors activate a series of signaling cascades that affect protein expression. A dysregulation of RTK signaling leads to cancer, such as CML. CML is presented when there are a growing number of myeloblasts found in the bone marrow and in the bloodstream. Myeloblasts are a genetic change occurring in an immature myeloid cell. Myeloid cells make up red blood cells, platelets, and white blood cells. When one of these cells changes and becomes a CML cell, it no longer matures the way it should. Often it divides to make new cells faster than normal. The genetic change that occurs in the cell forms an abnormal gene called the BCR-ABL fusion gene. The BCR-ABL is formed when pieces of chromosome 9 and 22 break off and trade places. The ABL gene from chromosome 9 joins to the BCR gene on 22 chromosome. It's also known as the Philadelphia chromosome. BCR-ABL fusion gene makes cells grow out of control. Here is a picture of a normal white blood cell count in the human body compared to that of an abnormal white blood cell count to those people who are affected by CML. These are the pathways in which CML occurs. Symptoms of CML include feeling of fullness, irregular heartbeat, fatigue, or lack of appetite. The areas affected by CML are the abdominal cavity and the bone marrow and bloodstream.